Hello. This lecture is called the Biochemistry of Autumn Tree Phenology. I'm not sure if you remember what phenology means from way back at the beginning of the year, but I'm going to keep reviewing you through things throughout this lecture, all of which will be on the midterm. So what factors affect autumn phenology? First of all, if you can write down in your own words what you think phenology means, do that somewhere on the slide. It's also has to do with losing leaves, which is like an aging process of a tree, which is called senescence, or senescence, I think it's pronounced. So go ahead and write down what you think phenology means, and push pause, and then I will tell you. So phenology looks at life cycles of living things. It has to do with how temperature affects those life cycles. So when I did a research project in Colorado way back at Rocky Mountain Biological Laboratory, I actually studied the phenology of a plant called Ligusticum porteri, which is a lot like Queen Anne's lace or Porter's lovage, which we have around here. The flowers look like, the bouquets of flowers look like kind of umbrellas, tops of umbrellas. And during that Colorado study, I was interested in how the blossoms changed from the top of the plant to the bottom, because essentially they were perfect flowers. They had male and female parts, and so you want to avoid, or the plant wants to avoid, self-fertilizing itself. So the timing of the flowers coming out had to do with how the plant would not cross-fertilize itself. So anyway, the factors that affect autumn phenology, if you probably could guess: sunlight, temperature, and storms. But what I want you to think more about and actually write down under sunlight is that, like, what are some things that would affect the amount of sunlight? So write down like 1 or 3 things below where it says sunlight, where you wrote it in on that line. Again, what are some of the things that would affect the amount of sunlight? Same with temperature. Write down temperature, but underneath that line, I want you to think about what are some things that would affect the temperature. And then storms, wind, and rain. Again, jot down what are some things that would affect storms. Now, you probably noticed in the picture, this is our study site. There's the classroom. We're at the upper window of the buildings. This is a really cool graph. So somewhere on this, I want you to write down, what is this graph showing? Really look at it. Look at all the labels. What is this graph showing besides sunlight, temperature, decline? So push pause and write that down, and then we'll talk about it. So this graph, I don't know if you noticed down at the bottom, is sunlight and temperature decline in Toronto, Canada. And the solid line is the amount of daylight, and the squiggly line is the temperature. And you'll notice there's kind of a delay in temperature from the amount of daylight, which is kind of cool. So as we get the shortest day of the year on the 21st in terms of winter, there's a delay of about, let's see here, 12 to 21st, so about 12 days till you get the hottest temperature, or likewise the coldest temperature. So why do you think there is that delay? Write that down somewhere on the slide, and then we'll talk about it. So what I'm going to start to get you to see is that when you talk about chemicals, that kind of leads you to organelles and, or molecules, then organelles, then cells, and cells will affect the overall organism. So these are leaf scars. I don't know if any of you, some of you have this tree. Notice the thorns. So this is the black locust. And so leaf scars are made when the leaf falls off. And if you can imagine, the tubes that run up the stem, those same tubes go all the way to the root. So there's one long tube made of cells which have cell walls that are stacked in order to give this rigidity, so you have the plant. So when the leaf scar is made, the connection from the root to the leaf is broken, and thus the leaf is falling off. So people can actually ID a tree also from leaf scars as well as thorns and different structures like that. Again, reviewing you for the midterm, take a look at this plant. Notice how the leaves, notice how they are arranged on this branch. Do you guys remember what that kind of formation or pattern is called from way back, one of the first labs you did? Write down the answer. We'll see if you get it right. You have the terminal bud. Remember, you had to know that for the study. There's the auxiliary bud. There's the leaf scar. Again, different trees have different shaped leaf scars. There's the node. Lenticel, that's where actually the tree can breathe through that structure. Remember we talked about the petiole. There it is. It's the part that sticks out of the leaf. And we did not include that in the measurement for our Harvard study. So what you need to be writing here is what type of pattern you think that is. We're going to start getting into kind of evolution. We're moving in that direction, genetics and evolution. So it's all about adaptation. How do these structures get chosen so that the organism lives and survives? So there's something called vascular tissue. It apparently evolved as an adaptation to life on land. It consists of a series of elements that form tubes, and they're represented by the blue lines throughout the plant. It is made up of 2 different kinds of tissues, and those tissues are called xylem, which is what the tubes that carry the water from the root, imagine it taking out of the soil, to the leaf, and it's going to do that for actually photosynthesis to happen, which we'll talk about later. And then the phloem is the tubes that come all the way from the leaf all the way down to the root, because you're going to make the sugar, the food, in that leaf, and then it's going to be moved from there. And so the phloem runs parallel to the xylem and carries sugars produced in the leaves. And these solutes are dissolved in fluid, that would be water, and that's moving these things to all parts of the plant. Note that the pattern of the veins we observe in leaves is actually vascular tissue branching through the leaves. So you can see here the blue lines. Those are showing you the vascular tissue, which are the tubes, and notice how they are running all the way down to the root. The destruction of chlorophyll, preparation for dormancy. So way back in the fall, I think we took our data until October 10th, I think it was, when most of the leaves had fallen off. I don't know if you guys noticed the pin oak over by the Quaker building still hasn't lost its leaves. So it'll be interesting to see what happens as the new ones come in. But here is chlorophyll. I want you to write down on this slide what type of format this chlorophyll is shown in. So write that anywhere off to the side. So as photoperiodism decreases, that's the amount of light, the chlorophyll stops being produced. And other pigments that were there all along, the anthocyanins, which are kind of reddish, the carotenes, which are kind of orangey, the tannins, which are brown, they were masked by the chlorophyll showing up. So there's so much chlorophyll, which is green, that it's masking the other colors. So when you stop producing that chlorophyll, because the temperature is dropping, and so the plant's not making it anymore, that's when you see the beautiful colors in the trees that we have up in New England. So to prevent damage from the cold, this is kind of interesting, the plant actually shifts from making chlorophyll to making sugars and amino acids, which both act as an antifreeze, because as you imagine, when it drops below 0 degrees Celsius, right, the water in the plant is going to start to freeze and expand and break the cells, so it needs to get rid of as much water as it can from the structures and make as much amino acid and sugar to stop it from freezing. So you'll learn more about chlorophyll when we get to photosynthesis. So why does the leaf fall? The nutrients and water are removed from the cell during that dormancy. So we're talking winter here. 2 types of cells exist at the petiole. So remember what the petiole is and where that petiole attaches to the stem. There are 2 different types of cells that are going to structure themselves depending on what they do. So there's the soft parenchyma cells, so that's on the leaf side, And then there's the waxy suberized cells, which is on the tree side. And eventually the vascular bundles, the veins, become really thin from all the molecules that are kind of moving out. And then the slightest storm can break the leaves and they fall, leaving a scar and then a bud, which is the next year's growth. So if you go out and look at our trees right now, you're going to see the buds waiting to come out as controlled by temperature. Now look at that cell. Okay, so what do you think? I want you to label the parts of this cell that you can label as part of the review. What's the blue? What do you think the white with the dots is? What do you think those little structures are? That there are tons of them. So go ahead and make your guess, write it down. And so parenchyma, the larger volume, nucleus is the same size, walls thins, vacuole developed, mature organelles, intercellular space. This cell is still isodiametric, so it's evenly made in the basic biochemistry of the filler cells. And last but not least, there is a really cool video down at the bottom here. Click on it. It's going to kind of run you through some more information. I want you to take notes as you listen to that video and learn about the autumn colors and the losing of leaves. And that is it.